So this is the second talk in this meeting from our lab, Molecular Pathology in ICGEB. And what I'll tell you, it's a little bit an overview of the role played by splicing factors in neurodegeneration. I'll be basically talking about one splicing factor in particular, and that is Tdp-43, probably the most famous nowadays. But I'll try to give you an idea of how finding out the involvement of this protein in neurodegeneration has opened up a huge range of new possibilities, both with regards to our understanding of disease, but also with regards to possible therapeutic options. So let me first of all give you a brief introduction, and just to give you an idea of how important it is to find out the molecules that are involved in these diseases. And basically, the story, I'll be talking basically about two diseases, and these are amyotrophic lateral sclerosis and frontotemporal dementia. So these two diseases have been known for a very long time, especially ALS. ALS was first described in 1874 by Charcot, who actually gave the medical definition of people who were affected by ALS. So people who had a chronic degenerative neurologic disease that was characterized by the selective involvement of the motor system. And this is a disease that normally, nowadays we know, is normally undergoes where there is the degeneration of the upper motor neurons in the cerebral cortex. The patients develop this disease usually when they are in their 50s or 60s, and within 3 or 5 years, this is an almost invariably fatal disease. It became very well known in the US in 1939 when this very famous baseball player, Lou Gehrig, actually died of this disease, and for quite a long time, ALS has also been known as Lou Gehrig disease. So the other disease that nobody knew was related was defined a little bit later in 1892, and the consensus was then eventually reached in the 1994, and this is frontotemporal lobar degeneration. Now, frontotemporal lobar degeneration is different from ALS because there is almost no, we'll see later that really there is, but it was initially described as not involving the motor neuron system, but basically involving the behavior. So people changed personality, they started to forget things, but the characteristics were quite different from Alzheimer's. And indeed, when people who were studying these two diseases actually compared their data, they saw that there was some similarity. First of all, there was some similarity at the phenotypic level. So this is actually drawing by Charcot. So you see, these people who had FTD and people who had ALS, eventually in the face, they started to have similar facial traits. Also, when you study for a long time people who have FTD, they were, recognizing that 50 to 20% of people who developed FTD were actually also developing some kind of motor neuron disease. And at the same time, it became clear that almost 50% of ALS patients actually present some kind of cognitive dysfunctions, especially at the executive, and they also showed some mild memory decline. So actually, 15% of all the people, of these 50% of ALS patients, eventually could actually develop a clinical diagnosis of FTD. So when people people were starting to looking for connection between these two diseases, and these connections were immediately very clear when they looked at the inclusions of at the time it wasn't known of what these inclusions were made, at the inclusions in the brain of these patients. So both patients who were affected by FTD and ALS, they presented some proteinaceous and ubiquitinated inclusions in various regions of the brain. and these look very similar. Both inclusions were negative for tau, that is one of the characteristics of people who are affected by Alzheimer, but at the time, and for quite a few years, almost 10 years or so, nobody knew what was the protein that was making these inclusions. Also, the genetics at the same time was very clear that these two diseases, who were apparently different from each other, were actually connected. Because if you look at large families who, find patients who have many children, you could see that in these families that were obviously at risk for this kind of neurodegeneration, the children could either develop ALS, pure ALS, or pure FTD. So this means that the genetics are actually connected with each other. And nowadays, what we know is that the protein that is occurring, that is aggregating in both kinds of diseases, is actually a protein that we have been studying for many years, and it's called Tdp-43. This was first described in 2006 by the laboratory of Virginia Lee in Pennsylvania. And what they actually did in this lab was to take human tissue from the patients, brain tissue from the patients. They separated the aggregates from the rest of the brain material. They used this aggregate to inject into mice. After injecting in mice, the mice, of course, were making antibodies against these aggregates, the proteins that were making up these aggregates, and they screened all the antibodies for the monoclonal antibodies for the ones that were capable of recognizing the inclusions. And after they found which antibody was actually recognizing the inclusion, then they went on to look what protein was actually recognized by the antibody. And the answer was Tdp-43. So a completely unknown protein, almost unknown protein at the time, because you see that until 2005, there were very few references to this protein in the literature, and most of these references come from Tito's lab. I was actually the one who kind of revived Tdp-43 in 2001 when I found that it was behaving as a splicing factor. But after 2005, the number of references to Tdp-43 have been doubling almost every year. And now I think there are around a steady 200 or 300 papers per year that contain as a keyword Tdp-43. So what happens to this protein, right? Now that we know the name, we can start to understand what is going on. So being a splicing factor, Tdp-43 normally likes to stay in the nucleus, right? This is where it does its major job. During the disease, for some reasons that are still not very clear, most of the protein that is in the nucleus is translocated to the cytoplasm, and in the cytoplasm it forms the aggregates that are cleaved to generate several proteolytic fragments, which are toxic to the cell, and also it's aberrantly ubiquitinated and aberrantly phosphorylated. And at the moment, really, the significance of this aberrant, the pathological significance of the aberrant phosphorylation is not so known. But when you have this information, right, you have a name, what you now have is the possibility to use this information to do many things. And this is what I'll be talking about in the next 20 minutes or so. So first of all, you can use this information as a biomarker. And we'll see why this is important. This is important because you can, of course, when you have a good biomarker, you can, of course, monitor the course of the disease. You could find ways of people who maybe are at risk of developing the disease much earlier before you see the symptoms. Because you see, the problem with all these neurodegenerative diseases is that because our brain is so good at making up for neuron loss, right? Once you actually realize that the patient has a neurological problem, most of the time this is too late to do anything, because a lot of neurons have already died, they cannot be replaced, and therefore it is very difficult then to treat in a medical manner the condition. So if you have a good biomarker, maybe you can realize that this guy is going to be sick much earlier than you normally would. Of course, a good biomarker is also very good when you test for new therapeutic strategies, right, to see if they're effective or not. But once you have a name, what you can do, you can try to model the pathological features, right, and in this way, if you model successfully enough, then what you can do is to use this knowledge to develop, again, new therapeutic strategies. You can, of course, understand its functions, and when you know the functions, then it's easier to, try some new ideas. And also, once you have a new name, you can screen for mutations. They don't occur very often in Tdp-43, but they do. And if you screen a population that you think is at risk for mutations in this protein, then again, you can use this information to find out, right, or at least realize which people you should look more carefully to see if they're going to develop the disease. So, in our lab, what I'm going to tell you in the next 20 minutes or so is something along all these lines to give you an overview of how many things one can do. And of course, the most important thing if you want to look for a therapy is to actually try to reproduce the aggregation process, because that is the main characteristic of the disease, Tdp-43, that becomes aggregated from the nucleus to the cytoplasm. And we now know that Tdp is a molecule that is very aggregation prone. I mean, it's not a surprise that you find Tdp-43 aggregated, because there are many, many things that can happen to this protein in the cellular life, such as excessive protein production, or cysteine oxidization, stress granule formation, cleavage, the presence of mutations, the faulty interaction with other HNRMP proteins or poly-Q repeats. So whenever some of these factors are becoming dysfunctional, then Tdp-43 will tend to aggregate either in the nucleus or in the cytoplasm. Now, as I said, there are many ways that you can induce Tdp-43 aggregation. But the problem until now, until 2 or 3 years ago, was that they were not very reproducible. I mean, you could just overexpress the protein in transient cells and you could see aggregation, but in one experiment you would get a lot of aggregation, in another one a little bit less. And so in Tito's lab, what we try to do is to find a way to reproducibly induce aggregation of Tdp-43 in a highly controlled manner. And the way we did this was actually to exploit a small sequence inside Tdp-43 that is characterized by the presence of many glutamine and asparagine repeats. And this is the core prion-like sequence that in many proteins is known to induce aggregation where it is found. And what we did was to simply take this very small part of Tdp-43 and repeat it many times after also joining it to an EGFP protein that would act as a reporter, so that we can easily follow what this protein is doing, what this effector is doing inside the cell. And you can see that when we added one of these repeats, then this is a filter assay to check for aggregation. When we added one repeat, there was really not very much effect. 4 also we could not see a lot. But when we added 12 QN repeats, then the protein started to aggregate on the filter. And what is more important, when this 12 per QN effector was transfected into cells, not only there was aggregation of this effector inside the cytoplasm of the cells, but also the endogenous Tdp-43 that would normally be basically soluble in a good proportion, in the soluble proportion. In the presence of these 12 QN repeat, the endogenous Tdp-43 was almost all trapped in these aggregates. And you can see it better here, where you see in green, you can see the EGFP 12/qN that is making all these small punctuate aggregates in the cytoplasm. But if you co-transfect the cells that are expressing this 12/qN with a flag Tdp-43, what you can see is that the 12/qN aggregates can actually trap the endogenous Tdp-43 and flag Tdp-43 inside the aggregates. And not only this effector can trap the Tdp-43 in the aggregates, but these aggregates also start to share some of the characteristics of the aggregates that are found in the patients. That is, they are ubiquitinated and they are also phosphorylated. So basically, in conclusion, what we now have available in the lab is a highly reproducible system to simulate the aggregation. And I'll come later on in the talk to show you how having this system can actually help to find new therapeutic molecules. So just another few words about biomarkers. We are just starting to be involved in this field, so we will soon start to also look at this issue. But basically, how can you use Tdp-43 as a biomarker? Well, what most people have done up to now is actually to see, because the brain is not very much accessible, is actually to start to see whether in patients, if you look at cerebrospinal fluid or even in the peripheral blood lymphocytes, whether the concentration of Tdp-43 is higher in the patients as opposed to the controls. And nothing much has been really solved until now, although there are some preliminary evidence that indeed, if you looked at people, for example, who ALS, and you compare it with people who have other kind of neurodegenerative diseases, then the levels of Tdp-43 that is present in the cerebrospinal fluid is rather increased with respect to the other neurodegenerative diseases. Of course, these results are not good enough to establish this methodology as a very accurate and reliable biomarker, but there is a lot of improvement also in this direction. For the search of mutations, again, right, people have been screening Tdp-43 in very large populations of patients affected by ALS, and now we know more than 40 or 50 mutations that they all cluster in the C-terminal region. For many of these mutations, most of these mutations are actually missense mutations, and for many of them, we actually don't know what they are doing. But what one can do is to actually use this information to see if some of them have a pathological significance. And very recently, what we have done is with people in Brescia, in the Spedali Riuniti of Brescia, what we have done was to screen a population not of people who are affected by ALS, but people who are affected by FTD, right, and for which it was not known that there were mutations in Tdp-43. And what we did find was actually a few individuals who carried mutations in Tdp-43, and you can see the list here. And we, for example, started to characterize the effect of these mutations by simply, for example, getting peripheral in lymphoblastoid cells from these patients. And you can see here, just by a simple Western blot, where apparently the mutation here of M359 to V wasn't, the appearance of Tdp-43 was not very different from the normal control. But if you look at another mutation that we found, N267S, then you see that also by Western blot and in these lymphoblastoid cells, the level of Tdp-43 expression was very much decreased with respect to the control. And this was actually true not just at the protein level, but it was true at the mRNA level. And so what we are now trying to do is actually to understand how these mutations work, right? Once you know the name, you look for the mutations, and then you can start to see what these mutations can mean in the context of the pathology. Then, of course, once you know the name, what is important is to look at the functions, and then look in the patients if these functions are altered in some way. So Tdp-43, as I said, is basically a splicing factor. We characterized it initially as a splicing factor, but now we know that, like many HNRMP proteins, it can do many things inside the cell. So it's involved in microRNA processing, in mRNA transport, instability for sure, and also there is some evidence that it's acting as a translational modulation factor. So, because we are a splicing laboratory, as I said in my first talk during this meeting, what we were most interested is to see what happens if during splicing, right? And one way of doing this, now one can do this in a very general manner, is to use microarrays. So basically, you knock down your protein inside cells, and using a microarray platform, you can see all the pre-mRNA splicing events that occur when the protein is present and when the protein is absent, and then you can see where the differences are. Because we had many tools at our disposal, we tried to do something that is a bit sophisticated than just simply knocking down the protein and see what are the changes. So we did knock down the protein, so we have normal cells, we knocked down the protein, but then we also had stable cell lines that where the protein expression could be recovered, and also where the protein expression could be recovered, but instead of expressing a wild-type Tdp-43, it was expressing a Tdp-43 that cannot bind RNA. And why did we use this mutant? Because basically, what we wanted to have is a data set, right? It's 3 data sets that could all be compared to the normal situation. So the first data set that came from cells where Tdp-43 was knocked down by siRNA, so simply looking what happens. Then the other data set was the splicing events that were recovered when Tdp-43 was restored inside the cells. And the last data set was the splicing events that were altered when Tdp-43 expression was recovered, but not with a wild type, with a mutant. And so the idea is because these microarrays give you several hundred results, our idea was to use these data sets to very much reduce the number of potentially positive hits. So we all were interested, out of all these splicing alterations, were the splicing alterations that were altered when Tdp-43 was depleted, that were recovered when we put back an SI-resistant Tdp-43, but which could not be recovered when we put back a SI-resistant Tdp-43 that could not bind RNA. And we ended up, I won't go very much into the detail, but we ended up with around 100 splicing events that, interestingly enough, if you look at the genes where these splicing events occur, they are all genes that actually affect the expression of other RNA binding proteins, that they affect the several interesting pathways from the point of view of neurodegeneration, like apoptosis. And so we are now in the process of characterizing all these splicing events. And I'll just give you one example. So, for example, that was completely unknown until we did this study. So this is a protein, actually, let me see if we can find, okay, one that is a little bit better. So this is a protein that, this MAD gene that is involved in apoptosis, Normally, in normal cells, you can see that this exon here, that's called exon 31, is normally more included inside the final mRNA than skipped. If you remove Tdp-43, then you can see that the level of inclusion is reduced with respect to the control. When you put back the SI-resistant wild-type Tdp-43, then you actually can recover inclusion of this exon. But if you put back a Tdp-43 that is mutated, that cannot bind RNA, and therefore cannot affect the splicing process, then you don't have any recovery in the exon 31 inclusion. And what this exon 31 does is actually it introduces a premature stop codon in the mRNA of the MAD gene, and as Lynn told you before, when you have this occurrence, then very often the pre-mRNA is degraded by NMD, and indeed, if you look at the levels of this mRNA, this is just what has been shown to occur. So, what about the therapeutic strategies, right? Once you have all these models and you know the function, you know the mod, you know that you have a model to do the aggregation, what can you do about therapeutic, right? And there are several ways. First of all, you have to decide what is the problem, right? Because when you have a protein that is normally nuclear and is aggregated in the cytoplasm, there are basically two possibilities, right? One is that the aggregates are toxic, right? So the problem is the formation of the aggregates in the cytoplasm, and this is called gain of function model. The other is that the aggregates may even be harmless or protective, but the problem is when you have the aggregates and you don't have any more protein in the nucleus, what is going on is that the processes that this protein is controlling in the nucleus then start to become dysfunctional. And this is called the loss of function model, right? And it's important, of course, both models can occur at the same time, but it's important if you want to look at a therapy, which one you want to concentrate. Because basically, if the problem is that the aggregates are toxic, right, then what you might be looking for is for molecules, compounds, or other proteins that could be able to disrupt the aggregates, right? I get rid of the aggregates, I get rid of the toxicity. On the other hand, if the aggregates are harmless, or mostly harmless, and the problem is that you don't have any more protein in the nucleus, then of course you can very well ignore the presence of the aggregates and find some ways of putting a more soluble, for example, Tdp-43 in the nucleus, right? So let me show you what has been done to tackle both possibilities. We have never done much for the gain of function therapeutic strategies, so I'll tell you the approaches that have been tried by other researchers in the field. So basically, if you think that the problem is really the presence of aggregates, what you can do is you can look for molecules that can disrupt, right, all the pathways that lead to the protein aggregation. So if you start from your soluble, nicely functional protein, what you could find is molecules that maybe inhibit the misfolding of these proteins, because these proteins, when they aggregate, they very often do that because they start to misfold. They don't. Also, you could disrupt the oligomerization. After you have the misfolding, then these proteins start to oligomerize. You could disrupt the fibril formation, right? That follows very soon after oligomerization. Or you can simply just to try find compounds who, even if you have the aggregates, who can promote or help the cells to get rid of the aggregates. And this has been done with a number of compounds, as I said. I won't give you any detail. And also, it has been tried in some animal model systems where TDP-43 has been increased very much, in which the expression of TDP-43 has increased much higher than normal levels, and in these zebrafish and C. elegans, it has been seen that increasing the concentration of TDP-43 is very toxic. And you can see that in some cases, like autophagy activators, they can actually rescue the overexpression of TDP-43 in these models. What we have concentrated in the lab is really something different. Is the loss of function, because what we think is that one of the major problems that is present in the people who have aggregates of TDP-43 is that there is not enough TDP-43 inside the nucleus to do its proper job. And so, together with Fabian Feguin in the Drosophila lab, it's now a few years that we have made a loss of function model of TDP-43 absence, and we have done this in Drosophila. And the way we did this is to get the Drosophila, look for the TDP-43, the endogenous TDP-43 gene of Drosophila, and I should say that TDP-43 is a very, very highly conserved protein, so that was very easy to find the homologue of TDP-43 in Drosophila. We knocked it out using some classical Drosophila genetic engineering techniques, and we looked what happened, right? From the phenotypic point of view, the flies that were not expressing TDP-43 were actually physically not very much different from the wild-type flies. But what was very different, and this is what gave us a good hope for this to actually model the disease, was that there were a lot of defects at the level of motor neuron of the neuromuscular junctions. So these flies, although from the point of view of the phenotype were not so different from the wild type, had very severe locomotory problems. And the reason why they had very severe locomotory problems was basically because their neuromuscular junctions, you can see here the wild-type fly, right? This is a typical neuromuscular junction of the wild-type fly. When you take away TDP-43, you can see that there is really a lot less junctions and butons, right, with respect to the wild type. And what is even more interesting from the point of view of using this model to mimic the disease is that you can see here, if you actually, instead of putting back the wild type Drosophila protein, you put back the human TDP-43, then you recover the original architecture of the neuromuscular junctions, and you also recover the motility, right? That is, with the human TDP-43, so this means that whatever we find in this model that is lacking the endogenous Drosophila protein will probably be also applicable to the human system, right? Because the function of these two proteins is highly conserved. This just to tell you. So basically, and here is almost the end of my talk, what do we want to do? What can we use? First of all, one has to understand how TDP-43 can actually, using our aggregation model, how these aggregates can spread from cell to cell. Because this is one of the possibilities that the disease in the patients can actually travel through these aggregates from one neuron to the next, and therefore, in this way, right, start from a single point and then spread in neighboring neurons. And now we can do this using our aggregation system, because we can actually start to prove this hypothesis that these prion-like sequences, like the one we use to induce the aggregation, could actually account for the spreading of the pathology between different cell and brain areas. Another thing that we can do, now that we know Tdp-43 is involved, what we can do is to start to understand how Tdp-43 can actually relate to other defects that are found in the brain of patients who eventually develop ALS and FTD. And in the case of both diseases in particular, not even just 2 years ago, what researchers have found is that not only there are problems at the level of Tdp-43, but many patients also present a 6-nucleotide, this 4G2C repeat expansion in one gene that whose function is still completely unknown, and that is called C9orf72. And these expansions, right, actually can go up into the normal people have 10 or 15 of these repeats, but after you start having 60 repeats of 4G2Cs, then what you start to have is, of course, a problem in the production of this C9orf72 protein that nobody knows what it's doing, but also you start to have the appearance of foci inside cells. And from the way these foci look, right, these foci are associated with Tdp-43 aggregation. So one of the things we are going to do and try to do using our aggregation model is to see, right, if you co-transfect with these C9orf72 repeats, if you can increase the amount of aggregates, or what is the connection between the two. And so basically, right, and I'll finish early, basically what happens is that once you find a new player in the neurodegenerative field, you can use this information in a huge variety of ways, right? So first of all, you can conclude, and now we know that not just Tdp-43 is involved, there are other splicing factors involved, that RNA metabolism, which was very often overlooked in the neurodegenerative field, is now becoming one of the fundamental players to understand why people develop ALS or FTD. Of course, you can, once you know the names, right, you can start to see what are the connections between Tdp-43 and all the other players, so to see which genes are mostly affected and which genes are mostly involved with neurodegeneration. You can look for mutations, right? So this will give you a better idea for the screening analysis. You can look for people who are at risk of developing the disease. And then, of course, you can see what other factors, and as I showed you before, there are many factors that can lead to Tdp-43 aggregation, what other factors are actually involved in changing a situation that is normally stable, like for 50 or 60 years, and this applies to both ALS and FTD, so people are perfectly normal, they play perfectly normal lives, and then suddenly in their 60s or their 50s or their 60s, they start to have these massive neurodegeneration processes and that lead to ALS or FTD. And so I just acknowledge the people who are involved. Fabien Feguin is the one in ICGEB who's working in Drosophila, and he has been of enormous help. Then, of course, is Tito, right, who has encouraged all of us to work very hard on this protein, and the other people in the lab, who Maurizio Budini and Laura De Conti, who managed to participate in the setting up of the aggregation model system, and Marco Barral as well. I'll thank you and I'll take questions.